All right, and we are live. Good evening, happy Tuesday to the CWD community watching from home. I am glad that you've decided to join us tonight. My name is Carrie Sparling and I'm CWD's editorial director, and I'm proud to be hosting tonight's pop-up Screenside Chat. We usually do them on Sundays, but we're rocking out on a Tuesday, and we're connecting with the folks from the DRI. So this afternoon, or this evening, I have Dr. Allison Bear and Sean Kramer joining me for this evening's discussion. And I know this is where it gets a little tiny bit awkward because they're sitting here on camera and you'll be able to see them in a minute, but I'm going to read their bios as though they are not here, which is an odd way to lead into something, but we'll just give a quick introduction. So Allison Bear is a research assistant professor in the Department of Microbiology and Immunology at the Diabetes Research Institute at the University of Miami Miller School of Medicine. And Sean Kramer is the Chief Executive Officer of the Diabetes Research Institute Foundation. That is a mouthful, sorry. He brings to the DRIF more than 2 decades of proven fundraising success and a track record in achieving outstanding results for capital campaigns and other major development initiatives. And I am thrilled that both of you are joining us this evening. So I'd love to have you say hello to the CWD community.
Hi, good evening.
Good evening.
And so I know we're kicking off with a presentation, Dr. Bear. You have a talk that you're going to be giving that's about 15 to 20 minutes. But I want to tell everybody at home to make sure as we are listening to Dr. Bear and Sean that you are leaving any questions that you have in the chat, which is on the right-hand side of your screen, or you can add them to the Q&A function and ask whatever you'd like as we're rolling along. Because I want to make sure that if you have questions, that this is like the best time to get them answered. We have these guys here right now live for you. So Dr. Bear, I'm going to give you the ability to share your screen. So
I think we'll let Sean, I think Sean has a little bit of an introduction before I get into the science part. So take it away, Sean.
Sure,
Perfect.
happy to. Thanks, Dr. Bear, and thanks, Carrie, and equally thank you to Jeff Hitchcock for allowing us to host tonight and to speak at the Fireside Chat, obviously on an off night. So everybody joining us, thanks for switching your days up. We appreciate that. We're excited to be here. And for those of you that are not familiar with the Diabetes Research Institute and the foundation, first and foremost, I will say that we are located in South Florida, specifically in Miami. And the DRIF and the DRI were started by an amazing group of parents who were focused on one thing, and that was simply to find a cure for type 1 diabetes. And that is exactly what we do. So I have one job in this world, and it's very simple, and that is to actually lose my job. So, you know, we are here to find a cure for this disease and to move on. And I'll just share a bit of my story. I actually am not just the CEO of the Diabetes Research Institute Foundation, but I'm also a patient at the DRI. So I was diagnosed with type 1 now about 5 years ago. And I am unfortunately the 4th generation in my family to be affected by diabetes, all the way up from my mom to my grandfather to my great-grandmother. And I am really tired of dealing with this disease, is what I would say. You know, I know a lot of you have lived with it much, much longer, but, you know, I have concerns in my family that my children are going to be impacted by this. So I'm appreciative and humbled and honored to do the work that I do. But really the stars of the show are the researchers who are, you know, behind the scenes working every day to find a cure. And we are the largest, certainly, you know, most comprehensive research facility in the country, with the ability to do all the way from bench research through to clinical research. Which is just amazing. And I invite anybody that's interested in coming down and actually seeing the building once we're back open from COVID. We welcome anybody that wants to come in and actually tour. It's an amazing place to come and see, and you actually get to see inside Dr. Bear's labs and through all of our researchers and the work that they're doing firsthand, which is just phenomenal. So I'm excited to have Dr. Allison Bear with us this evening. And for those of you that are going to be attending CWD in person in just a couple of weeks, she's going to be there, as will I. And her presentation is going to be on Thursday, I believe at noon, right, Dr. Bear?
That's correct.
Great, wonderful. Well, I am not the star of the show. The star of the show is Dr. Bear. So I'm going to turn it over to you to talk about the amazing work that you're doing.
Thank you so much, Sean. I want to thank everybody, as Sean has mentioned, and Carrie as well. We thank you for joining us on a Tuesday evening. And really my focus of the talk today is really immune-based therapies for autoimmune diabetes. So under the framework that Sean presented, really my lab, I'm a basic science researcher, so I'm one of those bench laboratories. So we ask biological questions using experimental models that we can't directly address in the patients themselves. So we will be talking about experimental models of autoimmune diabetes, and we will be focusing on what goes on in my laboratory, which is immune-based therapies. So again, I'm new to this audience. We don't have to go into so much detail about what is type 1 diabetes, but as you all know, the type 1 diabetes affects the beta cells in the eyelids of Langerhans within our pancreas. And that is mediated what we have by immune destruction. So in a healthy eyelid has a mixture of different types of cell types, including the beta cells that make the insulin. But in patients living with type 1 diabetes, the immune system has inadvertently started attacking these insulin producing beta cells. And therefore, this leaves the patient with an absence of these insulin producing cells. And so, therefore, this is why you have to take daily injections of insulin. But what is an immune response? So, just to make sure we're all on the same page, because as scientists, we love to talk a lot of details, but I want to make sure we're all on the same page. So when we think of an immune response, I like to give the example of what happens with skin wounding, because we all have experienced this in our lifetime. So it's a good example to get everyone on the same page. So we call this response inflammation. So when we think about the healthy skin, what happens? So I first want to let you know that when you think about your immune system, I want you to think of your immune cells as your own private army. So you have different kinds of cells, you have infiltrate, you have all the different things that you have to protect your body or to protect the land. So again, the immune system, you have a similar type of soldiers. So we have in a healthy skin, we have bacteria. We all have bacteria on the surface of our skin. And so just below the skin, we have these immune cells that really are the soldiers that sound the alarm. So they find the danger, they engage in the danger, and they let all the other immune cells know that there's danger. So we call these tissue resident immune cells, and they are the ones that sound the alarm for the danger. And of course, then throughout our circulation, we have other types of immune cells that are ready to hear the signals of the sound of the alarm and come to the area. So what happens when we get a wound on the skin? So for example, we get a splinter, which causes a wound in the skin. And so now these bacteria, these little green triangle things are rectangles, are bacteria. And so again, we have these tissue resident soldiers that are ready to sound the alarm and let all of these cells that are circulating in the body to come to this site of the wound. And so this response is called inflammation, and it's your first response of the immune system. So think about when you've gotten a wound on your skin, you might have some redness, some swelling, heat, and pain. And this is a sign of an active immune response that's occurring at the site of your wound. So again, you have all these different types of immune cells, these soldiers, and the first wave of soldiers that come in, and they're there to destroy the danger. And the danger in this case is the bacteria. So they're coming in, they're destroying the bacteria. And then we have a second wave of soldiers that come in that we call regulatory cells. So you have effector cells. These are the soldiers that are doing the destroying of the danger. But then you have regulatory cells that come in and let the effector cells know everything's okay. You don't need to keep destroying. The danger is gone, and please move on. So there's two ways of these soldiers that after the alarm gets sound. So once the immune response is all cleared, the danger is all cleared, the skin returns to its normal state, which is you just have these tissue resident soldiers, and you have circulating immune cells, which are ready to do the job when they're called to duty. So that's kind of an overall generalization of what happens with an immune response. So it's a balance of this effector cells and these regulatory cells. So you have this natural balance. And so this leads to a controlled immune response. But in patients with type 1 diabetes, there's a loss of this control in the sense that you have more of these effector cells that are causing destruction, in this case, to the insulin producing beta cells, to the cells that actually regulate. So instead of the danger being eliminated, these effector cells keep attacking. They keep destroying the insulin producing beta cells because of this misbalance. So really my lab focuses on how to restore this balance so that we can have a normal control of an immune response and stop that destruction of the insulin producing beta cells within the pancreas. So when we think about immune-based therapies, I'm a cellular immunologist, so I think about this morning, noon, and night. And so when we want to think about what are some of the key parameters that we need when we want to develop an immune-based therapy for the treatment of type 1 diabetes. So one is we have to inhibit those effector T cells, those immune cells that are causing all the destruction of the beta cells that we find within this islet. Second, we also need to think about a therapy that also encompasses stimulating the immune response, because remember, there's this misbalance. So we need to lower the effector response and raise the regulatory response. And by doing this, we want to promote beta cell health. So we want to take those insulin producing beta cells that may still exist, and we want to promote their health and fitness so that they can do their job. And we can also think about this when we think of transplantation. So some patients may get a kidney pancreas transplant or even an islet transplant. So the premise is still the same when we want to think about, we want to protect those new beta cells that have been transplanted to the patient. So the same principles will apply in that type of situation, as well as when we're talking about the autoimmune response or the destruction of the beta cells. So what is my lab really focusing on? So I would say over the last 12 years or so, we've been wanting to study a particular cell, which is called a regulatory T cell. So it's in that second wave of soldiers that come in and are trying to turn off the destructive response. So my lab has been studying these cells, and we wanted to understand the basic biology. How do these cells function? How do they suppress? How do they do their job? And how can we use them as a living drug? How can we use them to restore that effector and regulation to control a normal immune response? So that's what my lab has been focusing on. And one of the things we've been asking is, what is the environment that is important for these regulatory T cells? So why do we care about environment? So I like to tell people, we live in South Florida. The sun is shining most of the time. We love to go to the beach or hang out at the pool. And some may say, you know what I want to do for the day? I want to go to the beach. I want to work on my suntan. So what do you do to prepare for the day? You pack a chair, you get a cooler with some drinks and some snacks. Maybe you bring some music along, suntan lotion. So you're bringing all of what you need to accomplish your goal for the day with you to the beach. But just imagine now you have everything that you need, all the tools that you need, but you get to the beach and the sun is out, it's dark, it's night, or it's become cloudy. You can't do your job. You have all of the machinery that you need to do it, but the environment is not supporting the ability for you to accomplish your goal. So that's why we wanted to study the environment. What is the environment that these regulatory T cells need in order to do their job correctly? So that's what my lab has been focusing on, like I said, about the last 10 to 12 years. And so what we've done, what we've discovered, that if you think of this box here with all these different symbols, this is your immune cells. So this is your soldiers. And what we do is we have to manipulate or modify the immune cells so that we can give regulatory T cells that will protect the insulin producing beta cells. And that's kind of highlighted here in the pink hearts. So after we modify the immune system and we can give these cells as a living drug, as a therapy, and what we want is those regulatory cells to go to the pancreas and surround the islets and protect the beta cells and love up on them and just protect them from the effector cells that want to cause them harm. And so we have developed a therapy with this idea in mind. And so, as I said, I'm a basic science researcher, so we use experimental models to ask biological questions. So in this case, we use an animal model, which is called the NOD. So these mice get spontaneous diabetes, very similar to patients. So this is the most widely used experimental model to study the disease. And so when we use this experimental model and we use our Tregs, so what I hope you can appreciate, it's a little bit of a busy graph, but if you focus here around, this is percent diabetes free, you can see when we use T regulatory cells that can go to the beta cells and protect the beta cells, that we had NOD mice that had developed diabetes and we started our therapy. And when we do that, 100% of the mice had remained diabetes free up to 6 months. I should mention a laboratory mouse lives to be about 2 years old. So 6 months is a significant lifetime of the animal. And so this was very exciting to us. And of course, as scientists, we need to have all different controls to make sure that we're not causing harm, that we're just having therapeutic efficacy, and that the therapy is safe. So we performed other experiments. So in this case, because we had to manipulate the immune cells so that we could give these Treg cells that are going to love up on the beta cells, we wanted to make sure that these animals could still respond to normal danger signals. And so one of the most robust danger signals that we can get is in a transplant situation. So in this experiment, we took a piece of skin from one type of mouse to a different type of mouse. And because that's seen as foreign, not belonging, it causes a very strong danger response. And so those skin should be rejected. And so that's what this is showing here. So here you're seeing a healthy skin that's maintained, and you have the foreign skin that doesn't belong to the animal and it's being rejected. But most importantly, this tells us that these cells that have been treated with our Treg therapy, that they're able to respond normally to immune danger. When the soldiers that sound the alarm and cause an immune reaction to occur, this still occurs normally in these treated animals. So we really have reset the diabetes that was occurring in these animals, and we've cured them of their disease, and that they still are immune competent and can respond to normal danger. Because when we think about applying this therapy to patients in the clinical situation, of course, we want patients to be able to respond to the flu, just normal things. You get a cut on your skin. We want patients to still have normal immune responses. We just wanted to turn off the destructive response against the insulin producing beta cells. So where are we taking this research next? So now we know that these regulatory cells, they'll go up to the eyelid. So this is a picture here of an eyelid. This is using a microscope, and we use antibodies that can identify different types of cells, and they're labeled with fluorescence. So this is how we can see this signal. So this is an eyelid, and you can see here, these are the soldiers. These are the immune cells. They're coming into this eyelid, and they're attacking the beta cells to destroy them. So normally when we do this kind of research, it's on an experimental model where we've had to sacrifice the animal. And so this tissue is no longer alive. So it's just a snapshot of what was occurring at that time. So we call that a fixed slice. So this is the pancreas. We're looking at an eyelid. We're looking at the soldiers, but we can't really see how these yellow immune cells are interacting with the beta cells, with the eyelid, because this tissue is not alive. So what we're trying to do is now take our therapy where we can cure the diabetes in the treated animals, and we want to start to understand how these immune cells, these little circles that you see here, are all surrounding this eyelid. But the difference between the previous slide and this slide is this tissue is actually alive. So we now have the technology to remove the pancreas and get tissue slices and keep them alive. So this means we can study the interaction between the immune cells with the islets. And I have a little movie here. I hope it's going to work. Yes, so you can see these little flashes. So this is showing that these soldiers, they're active, they have a lot of activity, and they're interacting with the islet cells. So when we want to now do our Treg therapy, we can ask, how is our Treg therapy interacting with, how is it affecting this interaction that's normally occurring? How can we change it so that we have therapeutic benefit? So this is one of the things that we're getting ready to do with these living pancreas slices. So we can do these from the experimental mouse models, but we also have the capability of taking samples from human pancreas, from cadavers, from people who have died from an accident or things of that nature. We can also get living slices from a human pancreas as well. And so here this movie is just showing you that we can not only stimulate the immune cells, we can also stimulate the beta cells. So again, you're seeing these immune cells flashing, they have a lot of activity. And we can add something, you can see they just went really bright pink. And we basically stimulated the beta cells with a particular drug. So we can not only look at the interaction, but we can see how our therapy is affecting beta cell function and seeing if it's having positive outcomes. Is it improving it? Is it increasing beta cell mass? So those are all the kinds of questions that we can now ask that we haven't been able to ask in the past by using these living pancreas slices. So that's kind of where this research is going. And so we're very excited about it. And then the last project I want to talk about is a fairly new project in my lab. It's together with Dr. Francesco Vendrame, and it's really focused on this medication called dimethyl fumarate, or DMF, as a novel therapeutic agent for autoimmune diabetes. So why did we get interested in studying this medicine? So DMF has been used in Germany since 1994 to treat severe psoriasis. So psoriasis is another autoimmune disease, but it affects the skin rather than the insulin producing beta cells. And in 2013, this drug was approved by the FDA for the treatment of relapsing remitting multiple sclerosis, which is another type of autoimmune disease. And it does share several similarities to type 1 diabetes. And again, this drug was approved in 2013, and it is the most prescribed drug to these types of MS patients. So it's been given to thousands and thousands of MS patients since 2013. And it's a chronic therapy. They take it twice per day. So it has a known safety profile as well. And I don't want to get into all the different mechanism of action of what they described, what this drug does in the MS patients, but just to say that a lot of these mechanism of action that they see in the MS patients would also benefit patients that are living with diabetes. So we wanted to study whether this medicine could impact autoimmune diabetes. So again, we went back to our experimental model, and this is a diabetes reversal again. So these mice become diabetic, and then we start the therapy. So we gave these animals this drug every day for 12 weeks, and that's in the blue line. So these are the animals that received the DMF, and the black is the animals that were the control group. So you can see in the control group without treatment, these mice, their diabetes is not in reversal. They still maintain their diabetic state. But when we go to the animals that are treated with DMF, we can see around 70% of these animals that were treated with this medicine remain diabetes free for up to 6 months. And this even occurred after we removed the drug. So the animals were treated for 12 weeks, and then for an additional 12 weeks, they did not have the drug. And you can still see that these animals in the blue line still remain diabetes free. And we've also optimized this protocol to give a more regulated dose. And again, when we did that in a smaller number of animals, we saw that 100% of the animals remain diabetes free. So this is very exciting that a single drug treatment was able to reverse the diabetes in these animals. But what we also wanted to ask was what happens, can we prevent diabetes from occurring in this animal model? So we call that diabetes prevention. So these animals generally can start developing diabetes at any time around 12 weeks of age. So what we decided in the prevention study was to start treating animals at 10 weeks of age. And we did a similar thing that we did in diabetes reversal, which is we treated the animals for 12 weeks. So that's that red box and the dotted lines. So again, we had the black line shows the control or placebo group, and the line in blue is the animals that were treated with the DMF drug. And so you can see during the treatment, so within this red box, all of the animals that were receiving the DMF did not develop diabetes during this time, while the placebo or control group, we had around 60% of the animals that developed diabetes during this 12-week window. Again, similar to the reversal study, we continue to monitor animals when we stop the drug. And so now we're about 24 weeks after we've removed the drug. So 6 months later. So they got treatment for 12 weeks and now they've been about 6 months off of drug. And you can see we still have 50% of the animals that have not developed diabetes. And usually at this age time, we have around 85 to 90% of animals that usually develop diabetes by this age. So this is a pretty remarkable recovery of preventing the diabetes from developing in this animal model. And one of the things that we like to do to just kind of put it in perspective is we run a statistical test called a hazard ratio. And really what this statistical test told us that any time during the treatment period that DMF treated subjects are 85% less likely to develop diabetes than the control treated subjects. So we really are looking very forward to continue to move this project forward. And Biogen is the company that makes this drug for the MS patients. And we have been talking with Biogen in the hopes that they will support a clinical trial for type 1 patients. So that's kind of where this project stands at the moment. And that just kind of leads me to my acknowledgement slides. We couldn't do any of this work without the people in the lab that are coming to the DR every day. We really do love walking through the door every day and doing our research and asking questions and understanding how the immune system works in type 1 diabetes so that we can develop therapies. And of course, I have many collaborators that have contributed to this work, both at University of Miami and well as University of Florida. And then of course, none of this would be possible without the funding that we receive in the laboratory. So that's the end of my talk, and I will stop sharing. So I'm happy to take any questions at this time. And of course, you can also ask Sean questions as well. And we're here to answer whatever questions you might have.
Well, thank you so much, Dr. Bear. And I have some questions. So
Okay.
okay if I start with some of those. Sean, did you have anything you wanted to add before I jump into part of the Q&A?
Yeah, first of all, I want to thank Dr. Bear for her presentation. Every time I listen to our researchers, I think how much I should have paid more attention in science classes in high school. And so thanks for that. But I do have one question for you, Dr. Bear. In terms of the experiments that you're doing, obviously these are in mice. What do we think from a translational perspective, the expectation of how this is going to translate into humans?
So some of the most successful treatments we've seen in clinical trials with type 1 patients, for example, the anti-CD3. So they did that in recent onset patients, but they also last year, I believe the paper came out where they did a prevention study. And in both cases, they did have positive therapeutic outcomes in those trials. In the reversal studies, it only extended out to about, I believe it was almost 3 years before we started losing the benefit. And in the prevention study, they saw a 2-year delay in the development of diabetes. So that's pretty remarkable. And those anti-CD3 studies, those were initiated in this NOD mouse model. So we can learn a great deal of how the immune system, because that is an immune targeted therapy. So this model is a useful tool for us to understand how the immune response is occurring in autoimmune diabetes and developing a targeted therapy for patients that are living with diabetes. So we do see, of course, a mouse is not going to be exactly the same as a human. We definitely understand that. But at the same time, we can't go into the patients and take their pancreas out and go, hey, what's your immune system doing in there, right? So we have to find that balance of learning to understand the disease and I guess we would say biological complex situations that we can't address in the patients themselves, because let's be honest, you need your pancreas. So we do have to use these experimental models to address these types of questions. Did that clear it up, Sean?
That did. Thank you. Yes, I know there's always questions about, well, it's a mouse and when is it going to get into humans? And how
Right.
long is this? But, look, the reality is, as we know, is that science moves slowly, but it has to get
Yeah.
it right. So
Yes.
appreciate all of your efforts.
And also too, just as another correlative, I mean, the most advances we've had in cancer therapies over the last 10 years, we went, oh my gosh, I'm totally blanking on his name, but he got the Nobel Prize a few years, and that was 15 years of work in basic science and experimental mouse models to understand about checkpoint inhibitors. So these drugs are called checkpoint inhibitors for treating certain cancers, and they've had miraculous therapeutic outcomes in these patients. And that again came from very sophisticated studies that were performed in animal models to understand how biology works. So just as another, it's not diabetes, but again, in another immune targeted disease.
Well, I had a question, and actually it really is part of the CWD kind of community-based question, because so many people in our community are either raising children who have type 1 diabetes or are like me, an adult living with type 1. And so when you're talking about your research, how does your research play into the lives of people with established diabetes like myself? Or maybe not specifically me, but
Right.
for kids as well.
So definitely when we're thinking about asking biological questions, we realize the immune status of patients that have been living with the disease versus patients that maybe are at risk for developing the disease may be different. So when we are designing experimental questions and have our hypothesis, we are looking at both situations. So I would say, especially with our Treg therapy, where we see them go to the beta cells and they love all up on the beta cells, that was in a recently diabetic animal. So that would be, more in the honeymoon phase. And we haven't really haven't tried yet in animals that maybe had their diabetes longer and stuff, but we also want to study that therapy and prevention, which we haven't done yet, but we do want to look in diabetes prevention. But with the DMF drug, we're so excited about that drug because one, it's already an FDA approved drug. Two, it's already been given to thousands of MS patients. So it already has a known safety profile. And our preliminary data show great efficacy in both diabetes reversal. So people, in theory, people already living with the disease, and then people that might be at risk of developing the disease. So the clinical trial will probably be done in patients that already have the disease, have been recently diagnosed with the disease. But we are interested in doing a prevention first. But we do try to look at both situations because we know for the community, you're going to have both. So you want people that already have the disease. How are we going to develop therapies for those patients? But how are we going to also develop therapies for patients that are at risk for the disease?
Got it. And that's really good insight. Thank you for sharing that. And so this question is for both Dr. Bear and also for Sean. We've been talking about the word cure, that magic word, for a really long time. And it's hard to talk about it and not have it right there yet. So why do we keep looking? Do you guys think that we'll ever get there? Or how do you feel when you hear that word, and what kind of hope do you have involved?
Well, let Sean take a roll at it first, and then I'll give you the scientific view.
Yeah, I'm happy to. Every day we have researchers like Dr. Bear that are actively working on finding a cure. People like Chris Fraker, who himself is affected by type 1, Norma Kenyon, whose daughter has type 1. I'm obviously type 1. So we're in this fight to get to a cure. There's nobody that is walking into that building saying, well, I'm just not here to do what I'm here to do. So, I am very hopeful. It's why I came to the DRI. I really feel that it is the best place for us to find a cure, especially in light of what you hear from Dr. Bear and some of the other researchers that we have. And what I have said about the DRI is that we, in essence, have found a functional cure for diabetes, right, with islet cell transplantations. The challenge that you have with islet cell transplantation is that you have to take now the anti-rejection medication, which has this whole another set of problems and issues around it. So, if we can figure out the anti-rejection piece, which we're actively working on, if we can figure out other means and mechanisms, to get to a cure, that's what we're going to do. Our job is to uncover every stone and rock that we can to get to a cure. And, it's my mission to make sure that we get there. I often say, the history of the DRI or the DRIF, has been of late a little challenging. There have been a number of folks that have kind of sat in my seat. And, what I've said to that tune is, there have been a number of CEOs here, but I'm the last. And that's my commitment to, not just to the diabetes community, but to my children. We have to get to a cure, period.
Thank you. And Dr. Bear.
So I definitely feel that as technology keeps improving, I really think me and my fellow scientists, as I said, it's really a team effort. And one of the things I love about the DRI is we have a whole bunch of different brains all working on the same problem. So I'm a cellular immunologist, but we have bioengineers, we have geneticists, we have stem cell biologists. So we have all these different disciplines right there under the same roof. And we all work together because we can't be an expert in everything. But if we can find a different cellular source for these beta cells so that we can do transplantation in more recipients, we still would require immunosuppression, or we need to find ways that we wouldn't have to give it chronically, but still have therapeutic benefits. So my lab also works on that. And so we work together as a team, basically trying to solve each other's problems. And how do we balance all of the things that are going on in this very complicated disease? So just with that saying, and so I really do think technologies in many different disciplines has really taken off over the last 10 years. I mean, listen, we can sequence the entire human genome. Before, that would take months and months and months, and we can do it in a few days. That's pretty amazing. So I really think that technologies has really exploded over the last 10 years. And we've really learned a lot about the disease because we can ask many more questions that in the past we weren't able to answer. Like with these living pancreas slices, we don't know how these immune cells interact specifically with the islet cells. So are we going to have to target therapies that block that interaction? Or, just basic questions that we don't understand about the disease that we're now going to be able to answer. And when I like to have this saying, sometimes I do put it at the end of my talk, but it's not enough to do our best. We have to know what to do first before we can do our best. And as Sean said, basic science research takes time. Why? Because biology is complicated. Unfortunately, it makes my job fun because I get to make all these new discoveries, but it's complicated. So I really do think we're at a very important crossroads where we know enough about the disease, where we can really start impacting patients and moving therapies into the clinical situation. So I do see that there was a question in the chat about the trial. I wish I could tell you that the DMF trial is something that's already ongoing or taking participants, but we are still working in that direction to develop a clinical trial. So of course, if it becomes available, it will be on the DRI webpage. So you definitely should keep an eye out for that in the future. But I wish I could tell you you could sign up for that now, but we are really working on very hard to get that as a clinical trial going.
Yeah, and I would encourage folks, that if you haven't been to the DRI website, go to diabetesresearch.org. There is a place to sign up for the DRI Insider, and that will clue you into, the trials that we have going on, what new research is coming out. It's a really great resource and tool for folks.
Got it. Thank you for that. And I'll make sure I include a link in the chat to that page. But Dr. Bear, you had mentioned like 10 years ago, which is the perfect segue into another question that I wanted to ask you, which was about, aside from research regarding devices, management tools, that sort of thing, do you feel that there's a big difference in the research of today versus 10 years ago, that biological research that you talked about? Like, what would be
I
different?
definitely feel like we have made leaps and bounds. For example, on type 1 diabetes, we know the type of immune cell, what type of soldiers are doing most of the damage to the insulin producing beta cells, which we kind of had some clues 10 years ago, but now we really have definitive evidence. And not only do we know the type of cell, but T cells are part of this immune response that are very specific. So they express a receptor on their surface that makes them have a specific response. So we're starting to understand now, because we can do genetics and we can understand at the single cell level what this cell is, what kind of specificity is this going to have? Is there these T cells that they're all the same kind of specificity, or are they different in pools of different specificities? So these are questions that we haven't been, which we suspected 10 years ago, but we just didn't have the technology to really answer those questions. So we really are, I mean, leaps and bounds, I think, over the next 3 to 5 years about really understanding key questions that we need to develop very robust therapies.
That is so cool. Not to just break it down all the way, but that is just so cool to hear. And so another thing that I'd wanted to ask about, and it's very relevant because we're living through a pandemic and we are still currently living through a pandemic. And so during the course of COVID, we saw science move really quickly. Everyone across the world saw it. The vaccinations came up very quickly. Everything just seemed like it was moving really fast. And so we saw that science can get done really fast, if you want to call it that. Does this sort of model apply to diabetes research? Is there a way that things can speed up, so to speak, through what we care about here right now?
I think there is some ways it can be faster now with the newer technologies, but in regard to the COVID and kind of the speed of everything, especially when you come to the vaccines, that technology's already been there and it's been studied for already 10 plus years. So the knowledge was already there to move quickly. So what we need to do in type 1 diabetes is we have to get that knowledge so that we can use the new technologies that are now and coming in the future, that we can move the research quicker to the clinical situation. So yes, I'm hoping as we gain more knowledge of how the disease actually progresses, that we'll be able to get to a place where we're relatively speedy. Right? In scientific terms. Maybe Sean wants to add something to that, but
Only
yeah.
if he uses finger quotes. That's the only
I can do that. So, one of the things that I would say is during the pandemic, we adopted our kind of motto or theme was hope isn't quarantined, because we needed to make sure that the researchers were still able to do what they were doing. And they did that. I think the thing that we focused on from the foundation side is exactly that, right? Is that the entire scientific community came together to solve a problem. So what we have been asking ourselves in terms of the future of solving for type 1 is how can we be a facilitator and be a collaborator with not just equally from the foundation side, but from the science side. And so we're working closely with our scientists and doing a lot of outreach to other T1D organizations that are out there like CWD and others to talk about how do we all truly come together as one community, because I think there is an opportunity to push the science forward with everybody's help.
So, Sean, that was a great segue into another question that I wanted to ask you, which was about how much the DRI works with others who are doing diabetes research. What does that relationship look like?
We do. So we have a global federation that we are aligned with that are all researchers that are focused on, obviously, finding a cure. And so we do, I think, a great job from that perspective. But equally, that being said, this is an interesting space to be in. I've been in the nonprofit space my entire career, and have been at places like American Cancer Society and others, and have seen when a community really does come together and collaborates on the whole, what you're able to do. So there's still work to be done there, and I'm excited about kind of where the future is going to take us now post-COVID.
Got it. And Dr. Bayer, did you have anything you wanted to add to that?
Yeah, I mean, I would just say one of the things I think is a really strong aspect of working at the DRI, as I said, we have scientists that are working on different aspects of the disease, and we really do work together. So internally, we work together. But as you can see, I have a lot of working collaborations with University of Florida and other researchers around the world, from other scientists at the DRI. So we definitely reach out. We also have Alberto Puglisi. He's very much involved in NPOD, which is sponsored by the JDRF, with Mark Atkinson at UF. So, and of course, Jay Scholar was a head of TrialNet for a really long time, and Jay Scholar is still at the DRI. So we have many people that are still working and fanning out and trying to work together so we can really find a cure for the disease.
And I love the passion that you both speak about this with. It even translates through a Zoom screen, which is like it's a pretty good skill. And kind of jumping off from that. So Allison, or Dr. Bayer, Allison, when
That's
you
okay,
are,
that's fine.
sorry, I saw your cat in the background, so I just
I
went
know.
into name,
Yeah,
sorry.
you guys got to see my cat.
I'm just, like I said, glad my kids have not come through. But you as a researcher, like, so we talked about before the call had started, how much the CWD community, it ends up being this like tilled garden for the next up and comers in the diabetes community, whether they're advocates, whether they're researchers, whether they're the next nonprofit head or the person who makes a difference in some other way. We have a lot of those people who are dedicating their lives already at such a young age. So what would you say to some of these people who are up and coming about why you became a researcher and why they might want to become that too?
Well, I mean, I went to college because I always knew I wanted to do, well, I was always a science nerd. So I always knew I was going to be in the science field. And when I went to college, my junior year, I saw a talk from an outside professor that was visiting. My undergrad's marine biology. So this talk was about cartilaginous fish, so sharks, stingrays, and how you can give known carcinogens, so things that cause cancer. And cartilaginous fish don't get tumors. So that was my first introduction to immunology, and my spark just went from there. And I just knew that's what I wanted to do. I wanted to work in immunology, and I wanted to impact people's lives. I wanted them, if they had immune diseases, how can we fix it? How can we cure it? So I've always kind of had that passion for there. And, my parents were always like, just spread your wings and fly. If it makes you so happy and excited and passionate, just do it. So that's what I would say to the young people: follow your passion. I really enjoy coming to the lab every day. Science is probably 90% rejection or failure. We make hypotheses all the time. And I showed you all the things that worked, but I don't want to tell you about all the things that didn't work. But as scientists, when we try to design experiments, we try to design them in a way that if we go against our hypothesis, we still learn something, so that we can move on to the next question. So, and our grants get rejected, our papers get rejected. So there's a lot of rejection. So you really have to love it. And I, like you said, I do love it. It comes across, I do love it. So just follow your passion. If you want to go and do science, then just go for it. Go and do science. And we always have volunteers in the labs at the DRI, of course, once COVID's over. So again, young people can reach out and see about how they can volunteer for a summer in one of the labs, for example. So yeah, or just reach out to your local, university where you're from, and just see, do you take volunteers? How can I get involved? So I would say just do it.
I love that. And I know both of you will be in attendance in person in Orlando in a couple of weeks for Friends for Life 2021. And so, Sean, do you want to tell us a little bit about what you and Dr. Bayer will be talking about down at the session, and when that session is for folks who are going to be there in person, how they can find you and say hi?
Yeah, absolutely. So yes, so we will be live and in person. So we'll be at the Friends for Life booth, and we'll have a great booth set up. So I encourage everybody to come by. Dr. Bear speaking on Thursday at 12 o'clock. You'll hear her delve into what she just presented, and hopefully even more detail. And she'll be there live to answer your questions. And then certainly myself and the DRIF team will be present. I know a lot of our folks have met many people who have been to CWD for many years. I think we're one of the original sponsors of CWD, so we're excited to be there. excited to be back in person, certainly, and come on by the booth and say hello.
And I also just want to add to that, on Friday night at 7, there's like dessert with posters.
With the faculty, yeah.
I will be there with a very scientific poster, but I won't be talking so much into detail. But I will be there, I believe it's from 7 p.m. to 9 p.m. On Friday night. So again, that's another opportunity to just come by and hear more about the research in my lab or other research that's going on at the DRI. So feel free, that's another opportunity to come by and, talk science.
And the booth itself will actually have some fun activities. I was just reminded that we're gonna actually have a gaming theme at the booth. So if you have an interest in gaming, or we're seeing what we're doing in the gaming space, please, definitely come by and see us.
Awesome. Very, very cool. Well, thank you both so much for sharing your time with us this evening, again on a Tuesday. So thanks for that. And for everybody at home, thank you for joining us for this hour. This has been awesome. And I really appreciate both of you coming on. Is there anything that you'd want to add before we close up the chat this evening?
I just thank you to CWD and thank you, Dr. Bear. We appreciate you all, and thanks so much for spending some time with us. And we remain open and welcome to questions, comments, concerns that you all have. We're here. We serve the diabetes community. We're a part of it, and we look forward to seeing you hopefully in a couple of weeks.
Well, we thank you both so much for your time with us tonight. And for everybody at home, we're going to be having one more Screenside Chat before we close things up for the month of July, because we're going to take July off from these Zoom meetings. But the next chat is scheduled for June 27th at 7 p.m. and we'll be talking about glucagon. So we hope that you'll be able to join us for that. You can get more information on our chats and everything else that we have going on in the digital space at cwd.is/Screenside. So thanks everybody for joining us. This recording will be available in just a bit. Everybody waves at the end of a Zoom call, and we will see you all soon. Have a good night.
Bye, take care.
Thanks. Good night.
Good night.